So in this video, we are going to talk a little bit about vaccines, and specifically we're going to look at the different kinds of vaccines that are out there, talk about how they're produced and also when they might be best used. But before we get into vaccines specifically, we need to review a little bit of material and talk about antigens and how a vaccine is actually going to use an antigen to kind of fool the body into an immune response, making you immune to something. So antigen is actually a portmanteau. It stands for antibody generating, because an antigen is anything that gets into your body and it triggers an immune response. And when an immune response is triggered, you start generating, right? You start creating antibodies. So antigen is antibody generating. And as you've learned already, as we've gone through the immune system, an antigen can be many different things. So it can be a virus, it can be a bacterium, it can be a fungi, it can be other things as well, right? Anything that's going to stimulate the immune system. Probably the most famous antigen out there right now is the SARS-CoV-2 virus, because that is the virus that actually causes COVID-19. So I have drawn a little example of an antigen using the virus that causes COVID-19, because it's one that I think most people are familiar with. So as you look at my little representation of the COVID virus here, we've got this viral body, and then on the surface, kind of protruding out from that viral body, we have spike proteins, right? And when your body responds to COVID, it doesn't respond to the whole thing. Specifically, when your immune system gets involved, what it's responding to are these little spike proteins. So it's binding specifically to the spike proteins, and you have a little immune system cells in your body with a receptor that has the shape for that spike protein, and that's what initiates an immune response and causes antibodies to be generated or be produced in response to this antigen. The rest of this virus, okay, these parts in here are not necessarily antigenic to the body, so they're not what the immune system is going after, but they're necessary for the reproduction or the replication of this virus. So when the virus gets into your body, it's going to start reproducing using materials that are in here. And as it reproduces, it spreads, and that's what's actually going to make you sick. So what we need to do with any vaccine is we need to take the antigenic part of an antigen, so the part specifically that the immune system responds to, and that's what we are going to create a vaccine specifically for. So with that reintroduction to antigens and antibodies and those kinds of things, we're ready now to start talking about the different kinds of vaccines. And the first type of vaccine I want to talk about is what's known as an inactivated. I want to talk about this one first because it's probably the most simple and it's the kind that's been around for the longest. But what happens in an inactivated vaccine is you take your antigen, okay, so in this case we're talking about the COVID virus, and I'm not saying that there's an inactivated vaccine for COVID virus. There could be. There were like 200 developed. But what we would do if we were developing an inactivated vaccine for COVID virus is we would kill the virus, okay? Because if we kill the virus, it's not capable of replication, it's not capable of reproducing, and remember, if it can't replicate, if it can't reproduce in your body, it's not going to make you sick. So we kill the virus. I'm gonna give you this little picture, okay, because this guy is dead. And then we take that killed virus and we inject it into your body. And your immune system doesn't know, right? It's going to respond to the little spike protein just as if the virus was alive. The benefit of this is there's no way that you could get the disease from this particular type of vaccine, but you do get the immune response, which allows you to develop some immunity. One of the problems with this very, very simple type of vaccine is because the antigens are dead, it takes a lot of exposure to really get a strong immune response. So if you think about the polio injections that most of us got when we were children, that's an example of an inactivated vaccine. And the reason that you need 4 doses as a child is because we're injecting this dead microorganism, this dead virus, and because it's dead, we don't get a really strong immune response to it. So that's some of the cons of this particular vaccine type. So the second vaccine type I want to talk about is what's known as a live-attenuated. As the name implies, this virus, in this case, or the microorganism, whatever it is still alive, but it's been attenuated, which means that we have beat the snot out of it and it's severely weakened. So I'm gonna maybe draw this guy like this. He's not dead yet, but he's not doing well. And we've beat him up enough that he's not going to be able to reproduce in the body, right? So if he can't replicate, he's not going to be able to make you sick. So with a live attenuated vaccine, this is what we inject, not dead microorganism like we see with the inactivated, but actually a live microorganism that's really, really damaged and not capable of reproducing. This is going to give you a slightly better immune response than what you would see with an inactivated vaccine. However, there's a few cons to this one, one of which is it can't be given to people that have a weakened immune system, because if you have a weakened immune system, there is the potential that it would be able to reproduce in your body and actually cause you to become sick. So it's really limited as far as who can actually receive these types of vaccines. So the third type of vaccine I want to talk about is what's known as a subunit vaccine, sometimes also called a recombinant vaccine. But what's happening with this particular vaccine is we're not killing the pathogen, we're not weakening the pathogen. Instead, we're taking just a part, okay? So just a subunit of that microorganism, and specifically, we're going to get the part that's antigenic, and we're introducing a whole bunch of just that antigenic part into your body. One of the benefits of these is it does trigger a strong immune response, and there is no chance of it actually giving you the disease that it's meant to prevent, because, again, we're not including all of the stuff in the vaccine that is necessary for the virus to be able to reproduce. However, one of the problems with this vaccine type is it's really expensive and it takes a lot of time to make, because what you need to do in order to make it is you need to grow lots of your microorganism in a lab, and then you need to break it up, and then you need to take out just the part you want, right, which is just the antigenic part, and get that into a vaccine. So it takes a lot of time, it takes a lot of lab resources, it takes a lot of money in order to be able to do that, to produce enough vaccine to vaccinate an entire population. So a fourth type of vaccine is what's known as a toxoid vaccine. These are very, very specific for certain bacteria. So SARS is not a bacterium, it's a virus, but there are certain bacteria that are not antigenic in and of themselves, but they produce a toxin that when it gets into the body is antigenic. So obviously, again, SARS doesn't do this because it's a virus, but if you could imagine that this isn't SARS, it's actually a bacterium. Some bacteria produce, again, these substances that are toxic, and that's what a toxoid vaccine is actually going after. So in this case, what we do is we put just this portion into the vaccine. We're going to inject it, and your body's going to have an immune response specifically to that chemical that's produced by the bacterium. So a couple of examples of vaccines that we have that are made through this particular procedure would be diphtheria and tetanus, okay? Because diphtheria and tetanus in and of themselves are not antigenic, but they both produce chemicals that when they get into the body are antigenic and do cause the symptoms of those two diseases. So with this type of vaccine, what we're going after, what we're generating an immune response to, is specifically those toxins, so that if those microorganisms get introduced into your body, again at a future point, and they start producing their toxin, which would normally be antigenic and normally be harmful, your immune system's got it. It's gonna have a secondary response to it, and you're gonna have immunity to that particular disease. A fifth type of vaccine is what's known as a viral vector vaccine. So the AstraZeneca vaccine that was originally developed for COVID-19 was this particular type of vaccine. What happens with a viral vector vaccine is we take some other harmless virus that's out there, right? There's viruses that are harmful to the body, there are others that are completely harmless. So we take some other harmless virus that is out there, which I'm going to use that to represent, and then we take just the antigenic part of our harmful virus that we're trying to protect against, and we add it to our harmless virus. So with the AstraZeneca vaccine, what they did was they took a harmless virus and they added the antigenic part of COVID to it. And then they inject that into your body, and your body responds to just the antigenic part of it, and you get an immune response and you get antibodies being produced and you get immunity to COVID without actually being exposed to COVID and all the harmful things that come with COVID. Just as a side note, with the AstraZeneca vaccine, what they actually used was a virus that's found in chimps. So an adenovirus that comes from chimps, completely harmless virus. They knew it wasn't going to cause problems in humans, but the reason that they did that is because if they took a virus from chimps, they knew that the human population would not have been exposed to it. If they took a human virus that humans had been exposed to for years and years and years, many people would be allergic to it, which means that they would not respond to it because the body's already seen it and is going to recognize it as harmless. So it was important in this case to use a harmless virus, but also a harmless virus from a different species so that our immune system hadn't seen it before and would pay attention to those antigens and respond to it. So the last type of vaccine that I want to talk about are mRNA vaccines. And before we can really get into that, we probably need to review a little bit of cell biology to make sure that you're kind of understanding what's happening with this particular vaccine type. So here's my representation of a human cell. And you know that inside of a human cell, we have a structure which is known as the nucleus. And inside of the nucleus, we have DNA, right? Which is the genetic code for the cell. It's the blueprint for the cell. It's what tells the cell to do everything and make everything that it makes. So because of that, the DNA is really important, and it is protected within the nucleus, and it does not leave the nucleus. However, in order for the cell to be able to make stuff, we need to get information about the blueprint, okay, how to make the stuff, out into the cytosol and out into the organelles that are within the cytosol so that they can read the blueprint and make the stuff that the cell needs to make. So just normally in human cells, rather than moving our DNA out to where we're making stuff, right, proteins, what happens is we copy the DNA into what's known as mRNA. Okay, that stands for messenger RNA because it's a messenger from the nucleus to the organelles that are making stuff. And we release that messenger RNA into the cell. And then those structures in the cell, the organelles that are responsible for making proteins, they see that mRNA and they read the sequence, and the sequence of the mRNA tells them what proteins to make, and they make them, right? So that's how mRNA works, and that's how the cell works, and that's kind of some background that you need to have in mind as you're learning about these mRNA vaccines. So here's what we do with an mRNA vaccine. We figure out, okay, what the sequence is of nucleotides that's actually making the spike protein. And once we've got that sequence figured out, we make a whole bunch of them. And then we inject just the strand of mRNA that contains the sequence that tells cells to make that spike protein. We inject it into our cells. So it gets in there, your cell sees it. It doesn't know that it's viral mRNA. It doesn't know that it's not your RNA. What we see is between bacteria, viruses, plants, humans, all of the animals on the planet, we're all using the same nucleotides, the same genetic information. And so there's no difference. It's just making a protein of a different shape as far as your cell is concerned. So your cell sees this mRNA floating around in it. It's like, oh, hey, a message from the nucleus. Let's make what the nucleus is telling us to make. And so your cell starts to produce and release into the body the spike proteins, okay? And then the immune system doesn't know, hey, our own cells made this. It just knows I've got a receptor of the right shape to be able to bind to that. So those immune system cells that are active for the SARS-CoV virus are going to attach to that antigenic part that was made by your own body. So there's a lot of benefits to this, one of which obviously is we are not taking any type of substance that could potentially give you COVID and injecting it into your body. We're giving your body totally natural, what it already has, mRNA, that tells it how to make this spike protein, and your body's making the spike protein itself. And then That mRNA doesn't go into the nucleus, it doesn't change your DNA like a lot of people think actually happens. It's just there in the cell, it's used, and then it's broken down, and the nucleotides are recycled. Another benefit of this type of vaccine is, believe it or not, it's really simple and really quick and less expensive to make in a lab than a lot of other vaccine types. And here's why. We are really good at figuring out mRNA strands, and once we've got them figured out, then all we have to do is make a whole bunch of copies, and that goes into the vaccine. That is much faster, it takes less resources, and it takes less cost than actually growing out all of this virus and then putting that into vaccines, or putting parts of it into vaccines, breaking it down and putting that into vaccines. So it's very quick to make. It is very inexpensive to make compared to a lot of other types of vaccines, but it does have a con, and the con is because what you're injecting is mRNA, and mRNA is a fragile molecule. If it's not kept really cold, that mRNA will break down, and if it breaks down, you inject it into the body, the cells are not getting the full sequence, they have no idea what they're supposed to be making from this when it's broken down, and so it has to be kept very, very cold in order to work. So as I've kind of talked about these different types of vaccines, I hope that you have kind of started to think that there's no one-size-fits-all solution. For example, when we talked about this mRNA vaccine, it's really great in developed countries where you've got access to refrigeration. It can be made very quickly, it's inexpensive to make it, you can keep it cold until you can get it to people. But if you think about the situation in maybe a developing country where they don't have access to refrigeration, an mRNA vaccine is not going to work there. But a viral vector vaccine would, because it's not going to have to be kept cold. So that's why when we first went into the COVID pandemic, we had, I think it was something like 200 different types of vaccines that were in development to see what would work best, or what combination of vaccines would work best to serve the world's population. And that, in a nutshell, is vaccines and the different types of vaccines.